Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Janine La Salle from University of California, Davis on this show. Please let me briefly introduce you to our audience. You got your PhD in immunology from Harvard University in 1993. You then moved on to do a postdoc with Mark Lalonde at the Howard Hughes Medical Institute, and you joined the Genome Center at the University of California, Davis in 2000. And you're still there today. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Yeah, probably my parents and my upbringing had something to do with that. My father was a physicist and my mom's a public health nurse. But yeah, it started out, I was born when my dad was in grad school. So we went from living in graduate student housing to moving to a 32-acre property in Virginia, which was like a small farm. He got a job working for the government. This Cold War times. So there were woods to grow up in a big garden animal. We had sort of experimental animals. So I learned a lot about sort of nature and at that level. But then, I got interested more in the medical side. My dad unfortunately died of a brain tumor when I was 12. So that got me interested in medicine, and my mom kind of pushing me in that direction. But then I get to college and I kind of got pushed. I think I went to a small liberal arts college, and my professors tended to push me more towards the research. They saw how I was in the lab and said, you'd be a good experimentalist. So they encouraged me to apply for summer undergraduate research programs I went to Medical University of South Carolina in Charleston for 2 summers. And then that got me interested in immunology. So I applied for graduate school in immunology and ended up at Harvard, where there I migrated to a lab working on multiple sclerosis for my PhD. And then around that time, I met Mark Lalonde towards the end of my dissertation work, and he's the one who converted me to go into epigenetics. One day, we were collaborating with him on growing T cells, and he actually came to the lab to learn how to grow the T cells with me. And at one point said, I've got a postdoc, and he was a Howard Hughes investigator. So it was a great opportunity. And he's the one who converted me to work on this strange locus of Angelman and Prader-Willi syndrome. So that's kind of how I've sort of started with interdisciplinary biological background and ended up in epigenetics.
So this was around the turn of the century, right? So
yeah, it was 90s. Well, I started my postdoc in 93. So this is around when, I'd like to say that in those days, epigenetics was kind of the quirky exception to Mendelian rules, right? So people working on imprinting and exon activation. And it was always of interest to the human geneticist, but it was kind of like that, there's the weird folks that work on the things that, are odd.
That would have been my question. What was the field of epigenetics like back in those days?
It was pretty much imprinting and exon activation. And again, it was always interesting. I think people were interested in what we were working on, but nobody thought it would apply to, all sort of more complex diseases other than these rare ones.
So coming to your science that centers around genomic imprinting, human autism spectrum disorders, and the methyl CpG binding protein MECP2, a PubMed search with your name yields around let's say 137 publications. It could be more. Obviously, we can't go through all of them. So let's see if we can talk about the most important things. I want to start in 2013. There a paper was published in PNAS titled The Human Placenta Methylome. Can you talk about this study and what you found?
Yeah, so this is the early days when we started to get interested in, sequencing based for looking at DNA methylation patterns. And my postdoc at the time, Diane Schrader, was really interested. We had done just a little bit of pilot experiments on whole methylome WGBS, or whole genome bisulfite sequencing, following up on the Lister and Ecker paper, which again, I think when that paper came out in 2009 in Nature, that to me was like the first satellite images of what the methylome looked like. And they had seen, it's kind of buried in their paper, but when comparing like IMR90s, which is a fetal fibroblast line, to embryonic stem cells, they'd seen this phenomenon called partially methylated domains. And we got kind of interested in that because with our first sequencing of like a neuronal cell line, we'd seen something that looked like that. And Diane really had the idea. It's like, well, what about placenta? We know it's hypomethylated, but nobody knew if there were actually partially methylated domains. And in the past, it'd really only been seen in tumors, and like these, fetal fibroblast line, which is kind of grows like a tumor. So yeah, she decided to look at the placenta, and we collaborated with folks to get us placenta samples. And again, it was really a striking landscape. If you look at there's one figure in the paper where it just really drops down. There's these valleys of hypomethylation that are the partially methylated domains. So the placenta really has this unique landscape of partial methylation, and those regions actually have higher. Well, it's the regions that are partially methylated are actually go along with gene silencing. So it's kind of the opposite world in the placenta, where the high methylation domains are where the active genes are, and the partially methylated domains are where the genes that tend to be silent, although they can become activated. Later on, we also published to figure out, why the placental methylone could be useful, and then we did a comparative study across a bunch of different mammals. And learned that, all mammals' placentas are really different. It's a very bizarre tissue and recently evolved. But what they all had in common was hypermethylation over the gene bodies was what you see as actually a predictor of gene expression. So again, that's where it's sort of opposite world. It's actually higher methylation goes along with gene expression in the placenta.
So placenta seems to be like a non-canonical or a non-usual model system. So what makes it so interesting or such a good tissue to study?
Yeah, I think, to me, it's like for human disease, again, we're like in autism spectrum disorders, we're always looking. I mean, we've done a lot of work in the past on looking at postmortem brain, because obviously brain is where you think the epigenetic action would be. But clearly you can't diagnose someone from their brain. So there's been a real desire in the field to find a surrogate tissue that you can use. And a lot of people have used blood, but it's, blood after a child's been diagnosed with autism. But we really were thinking about the perinatal period as the time when that's where the environmental exposures anyhow are predicted to be in autism are predicted to act, as well as the genes themselves. A lot of the autism candidate genes are ones that are expressed in fetal brain. So you can think of the placenta as like a time capsule of what the fetus was exposed to and those gene environment interactions that occurred in utero. So that's what makes it unique. It's a very understudied tissue, and then somewhat ignored. It's, considered after birth. It's afterthought. It gets thrown away in the, if you've been in a delivery room, is usually like a bucket that collects the placenta and cord, and it gets thrown out with the trash. So for these kind of studies, we really have to collaborate with epidemiologists who have a whole team of people there ready to go into the delivery room to get those samples. So that it's definitely not tissue that's, again, normally disposed of, but it's not that actually easy to collect usually, unless they're being routinely collected for research study.
So there is an epigenetic or a connection between the epigenetic signature of the placenta to the embryo then?
Yeah, so that's what we've really been working on a series of papers, but that the placenta actually can reflect much of what's going on in the developing brain. Potentially other tissues too, but again, the brain is really the one that is the hardest to build in life, and probably it's especially in primates, where you've got to build this big brain with social and language, and so yes, it's what we've been able to establish is the signature of the placenta can often reflect, if we overlap the differentially methylated regions, we see a significant overlap between what we find in placenta and what we find in brain. And we've seen this both in the human samples, in primates, as well as in a mouse model of exposure.
Yeah, so you also looked at the evolutionary conservation of DNA methylation patterns and transcriptional regulatory programs in the placenta. You then performed the genome-wide methylome, methyl-C-seq analysis of human rhesus macaques and a bunch of other animals, placentas, as well as opossum also. So what was the motivation behind that, and what did you find then?
Yeah, that was really to understand why the placenta, what was the convergent aspect of the placental methylome among mammals. And it was striking because if you look at like the global methylation levels, there are big differences. Cow had like very low methylation. And but when you line things up, especially when we line like the human versus the rhesus methylation, even though the global methylation was slightly different, you could see the same repeating pattern of the partially methylated domains and the higher methylated domains. And again, what we were able to determine was what's really conserved is not so much, the height of the landscape or even where, the presence of clearly defined partially methylated domains, but it's the higher methylation over gene bodies that determines methylation. So, and that kind of led us to, well, maybe we could use this as sort of a way of measuring. It's kind of like carbon dating of what happened. So what happened in placenta, you almost have these footprints of differential methylation that could be laid down by differences in expression that could have happened very early in development, but they're still there in the term placenta when we collect it.
So even though it's all like mammals, there were like huge differences between all of them, and like the concept was the same, but like the amount and the signature of the methylated regions were very different.
Yeah, really. Right. So I think absolute methylation is less meaningful than the relative methylation. So the gene body versus the region around it. So when you're in a partially methylated domain, it's overall low methylation. But if you have a gene that's getting expressed, you'll see a higher methylation over that. And we could actually use that as a predictor for genes that were being expressed in the placenta.
What you then investigated was the placental methylone with respect to autism and it being a potential predictive biomarker. So what did you find there and how did you move into that? Or was this like the obvious choice or the obvious path to go?
Yeah, it was what I call a lesson in patience, because I think from the time we had the concept of using the placenta methylome to find something and then getting enough samples, right? So the paper we published in 2022 in Genobiology was the one that kind of paid off that the patients. We finally got enough samples to really get a strong signal so we were looking for differentially methylated regions with placenta samples for kids that either went on to develop autism or not. And so in order to do that, we actually took advantage of a high risk cohort. So with autism, there's about 1% to 2% in the general population, but in families where they have one child with autism, the chance of the second child having it is in this cohort was about 22%. So about 1 of every 5 placentas that were collected in this study would have autism. Because we were sequencing, it was rather expensive. We waited for the autism diagnosis at age 3 from this longitudinal study, and then went back and got enough samples to compare the two. So basically, we're looking at risk for autism before it ever develops, right? So in the newborn. So we saw this nice signal. It's rare in in this field to be able to see something that's of genome-wide significance, but there was this whole block of methylation difference on this region of chromosome 22q. And it was a region where there's really not any known genes in there. So, but we started hunting a little bit more, and it's definitely a partially methylated domain. We hunted a little bit more, and there was at least one transcript that was right kind of in our block. And it was just, one of those, the LOC with a bunch of numbers after it that had been described as expressed in testes, but really nothing beyond that. So we decided to look into that a little more and did some functional analysis of what that gene is and how it's being expressed. And it turns out it actually is, again, it goes along with the methylation. So in autism samples, we saw lower methylation of this locus that went along with lower expression of this gene. And then we did some functional studies to figure out what it does. And it turns out it responds to, we saw it go up with neuronal differentiation and with hypoxia. So hypoxia is really important for the placenta and the fetal brain. It actually gives the placenta a signal to keep going. And again, placenta is very similar to cancer in that way, that it has to do angiogenesis. And when it's getting hypoxic signals, it gets a signal to divide and find new blood vessels. And actually, we did see when we overexpress this gene in HEK cells, we saw an increase in proliferation. We also found a little peptide. So it was, annotated as a non-coding RNA, but sometimes the non-coding RNAs can actually encode peptides. And so we found this 20 amino acid peptide that we made an antibody to it, and it looks nuclear. We're still trying to figure out what it does, but we have this idea that it may be neuroprotective in response to hypoxia. So that's an angle we're going now. We also found a genetic link. So even that we were looking for the epigenetics, but a lot of times, as you know, with epigenetics, these EWA studies, you could actually find a genetics. And there's a big insertion. It's a 1.7 kb insertion nearby this gene. And it's very common in the population, but it actually wasn't in the reference sequence. So it's, again, been ignored, but it's there. If you look at like NOMAD database, it's there. And it's actually, 70% of the population has one copy of this, and about 10 to 15 has two copies. And it's the ones that have two copies that were the risk for autism. But then there was also an association with prenatal vitamin use, which is, so most people know prenatal vitamins are encouraged for women to prevent neural tube defects, but they're also protected for autism if taken in that first month of pregnancy. And so it turns out that the ones that in this cohort, if the women were not taking the prenatal vitamin and the child had the two copies of this risk allele, that was the highest likelihood of autism. So you could maybe protect, and we think again the folate and the prenatal vitamin are counteracting a lot of the oxidative stress reactions. So we're excited about this. It's a new gene, new locus. It's a tricky region of the genome to work in, which is probably why it hasn't been discovered. It's kind of those dark areas that are still in the human genome that are left to be undiscovered. But so finding good genotyping assays, and again, when we're starting with a new gene, there are no reagents in this locus. So it's fun, but it's challenging.
So up until now, we only talked about DNA methylation, but have you also looked at other epigenetic markers?
We have. I mean, I think we specialize more in DNA methylation, but we always look at that as a reflection of the chromatin. So we do chromatin immunoprecipitation sometimes for like MECP2, for histone marks. We've done a little bit of ATAC-seq, and we're starting to go more single cell. Right now, we're looking mostly at single cell RNA-seq, but we'd like to get into the single cell epigenetic assays and through collaborations and things like that. So we are starting to think about single cell methylomes as well.
So in the last couple of years, like liquid biopsies have become more and more popular. So a liquid biopsy that would be possible in this like area is like cord blood. Can this also be used? Have you tried that?
Yeah. So in the same samples from this high risk cohort, we had placenta and cord blood, and it's interesting because we also did the epigenome-wide study of the cord blood, and we found a different signal there. I mean, it was less of a sort of one hit on one chromosome, but the signal we got, even though this is blood, when we looked at the GO terms of the differentially methylated regions, they were really enriched for early neuronal development, as well as enriched on the X chromosome. So that's also got us thinking about, again, there's a strong male to female bias in autism. It's about 3-to-1, and it's really poorly understood. It's known that, people talk about females having the female protective effect against many neurodevelopmental disorders and as well as in utero insults. So there's something going on in the X chromosome. And, we think maybe we got this gene exact, which is a primate specific one that's sort of counteracts exist. And so we're thinking that maybe what we're seeing in the cord is actually some reflection of X chromosome erosion that's been implicated with exact. And there could be, again, environmental or stochastic reasons why some early embryos would have X chromosome erosion. And it's probably just subtle enough that obviously the fetus survives, but it could be involved. There's a lot of, the X chromosome is enriched for neurodevelopmental genes already. So anything, even subtle, expression changes on the X could potentially increase risk for autism. Mm. So yeah, and it's interesting because if you think about the early embryo, you've got the trophectoderm, the layer of cells around that becomes the placenta. So it's a very different lineage from the embryo itself, which comes from the inner cell mass. And so I think it's interesting that even from the same individuals, we're seeing very different epigenetic signatures, whether it comes from the placenta or whether it comes from the blood. And I think they're telling us different things that are both very interesting.
Yeah, because the placenta is more the embryo and the blood is more the mother, or what is the connection there?
No, I think the cord blood is, they're both from the fetus, really. We check for maternal contamination, but it's always pretty low, less than like 2 to 5% or something. So yeah, both are from the embryo, but they come from the two different layers, either the trophectoderm, which comes the placenta, or the embryo, which is all the tissues in the fetus.
So just last year, so we are recording this in 2023, so just if people listen to this later, you and your team reported the effects of prenatal exposure to a human relevant mixture of PCBs on the DNA methylation profiles of mouse placenta and fetal brain. So, I mean, obviously the exposome or the things you're exposed to are very important for your development. So what did you find in this study?
Yeah. So again, that's another one that fits into sort of the placental brain connection. So what we did, so there we were, and it was interesting. Again, this is the same cohort called the Marble study where, so what it's human relevant in that they measured the PCBs in the women in the study and then mimic this in a mouse model. So this is my collaborator, Pam Lyne's lab. And so you give this to the mice perinatally. And then we took, is in utero, really. And then we took E18 placenta and brain. And strikingly, when we looked at the methylome, there was, again, a really nice overlap between placenta and brain. So when we look at the placenta samples, we actually see overlap with, it was mostly neuronal go terms. And interestingly, too, because again, we also have an interest in MECP2, the pathways that were affected, if you look at enrichment, you can compare to all the different RNA-seq studies that have been out there. And MECP2 mouse models were like one of so the genes that are dysregulated in the MECP2 mouse models had a strong overlap with the ones we were seeing with the PCB exposure. So again, the idea that there are convergent pathways between, genetic causes and the environmental causes that are both affecting neurodevelopment.
So you also discovered a novel autism gene for epigenome-wide from epigenome-wide association studies of the placenta. So was this the one that you talked about?
Yeah. So this is we called it NHIP for neural hypoxia inducible placenta associated. So we got to rename. This is that one where we found it's a LOC number, and we got to rename it based on the functional studies that we did. Yeah. So that's the NHIP on chromosome 20.
Is there more to that, as you already shared?
Well, we just have a new grant starting, so we're starting to ask the questions. Yes. And it's really just functional. Again, when you get a new gene, you have to figure out what it does. So the really question is, how is it acting? We think it's acting as it's primate specific. And it's only 20. Yeah, the peptide itself is only 22 amino acids. And the last 9 amino acids seem to be mimicking transcriptional activation domains. So we think that it may be acting as a mimic and inhibiting protein-protein interactions. Again, there's a response usually in response to oxidative stress or hypoxia to sort of slow down cellular metabolism, slow down translation. So we think that's where our working model is right now. But yeah, we're asking, what does it do? What are the protein partners? How is it regulated? Again, the promoter is really unknown, but we think it's going to probably have this hypoxia response element. And also just, is it neuroprotective? So we've started doing some cell culture experiments, adding it to see if it inhibiting reactive oxygen species in the neuronal cultures. But we're also going to try injecting it directly into mouse embryos. Mouse doesn't have this gene normally, but again, and we think that the pathways that it's regulating are conserved. So that'll be interesting to see.
Yeah, sounds very interesting. So another buzzword in the last couple of years is really multiomics. And you also did a study last year that utilized integrative multiomics to examine maternal obesity effects on offspring neurodevelopment in rhesus macaques, again, by comparison to lean controls and to interventions. So what was the experimental setup and what did you find then?
Yeah, this was another collaborative study we did with some folks at the Primate Center, as well as an obstetrician who was really interested in the maternal obesity. So as you probably know, this is, a big problem now, that just the level of obesity and overweight has increased in the population. And it's about severe obesity causes about 1.8-fold increased risk of autism. And so, but again, it's really hard to study in humans because there's so many other things involved, and clearly it's not a risk factor on its own. So we went to the macaque model. So in the indoor rhesus macaques, some of them naturally get obese. So they could choose the female dams from the ones that were either obese or controls. And they also did 2 interventions. So both a caloric restriction, meaning they just got less food each day, and then a statin, a pravastatin drug, which is reducing inflammation during pregnancy from the obesity. And so the great thing about this, which is something we can never do in humans, is we got access to, the placenta, but also, but you can't get the term placenta from monkeys because there's interesting reasons for that. But what we were able to do, they took blood from the dams for like 4 time points during the pregnancy. And they were already looking at the cell-free fetal DNA because they wanted to, with monkeys, they're very expensive and they wanted to limit to a single sex to reduce the sex variability. So they were already testing for the Y chromosome. And we're like, well, we can use that for the methylome reflecting the placenta. And that actually worked better than placenta biopsies that they were trying to take. So we got cell-free fetal DNA at 4 different time points during the pregnancy. And then they did a lot of behavior so they could measure really cool social behavior in these monkeys. And then they took the brains at 6 months of age. So we got 3 brain regions that we could compare to the cell-free fetal DNA. So again, unlike in the human, where we never get the brain from the same individual as we can get the placenta, in this case, we could get the cell-free fetal DNA that matched the brain. And it was remarkable, again, how well it could, we found a couple of interesting regions and really the interventions were kind of in between. So, we saw regions that had differences for the obese and the control, with the interventions being kind of intermediate. And we also kind of looked at multiple genes that were part of this DUX4 complex, and that also kind of followed that. But what then we were also able to do with that is then our other collaborators were doing the immune system, so looking at a bunch of cytokines for the inflammation which occurs during obesity, and then metabolomics for both lipids and all types of metabolites. So we did these really big comparisons of associations with everything. It's kind of an everything by everything panel. And it really does help then kind of tease out what the important components are. And I'm excited about the future for cell-free fetal DNA. Clearly, it's being used in the clinic now for genetic testing, but there could be epigenetic tests as well that could be combined potentially with other biomarkers.
Yeah, cell-free DNA is really also the new kid on the block that is really interesting also for cancer and things like that. So again, something that is used all over the place.
Yeah, I think the cancer field are really more advanced on this, and we're learning from them. We actually are starting to do some interesting cancer. Again, right now, where we're looking at epigenetic predictors of disease, a lot of things come down to the same. So there's overlap between autism and cancer and a lot of the genetics. And I'm working now with 3-generation cohort, which is really interesting. This is Barbara Cohn, who's an epidemiologist in Berkeley. And she started saving samples in the 50s and 60s in Oakland, anyone who came to Oakland Kaiser Hospital. So it's a nice diverse population and interesting exposures, because these were the ones working in the shipyards in the 50s and 60s, where there were a lot of chemical exposures. And so they've been following, there was like 20,000 births they collected, and you can go back and then try to match the F1 and F2. So we now are working on an autism study with the F2. We can get newborn blood spots from these same families. And we're starting to also look at cancer risk, because they're interested in cancer risk in the women that are now in like their 50s and 60s and getting cancer. So we're hoping to actually maybe inform some of those cell-free DNA samples. Potentially we can get from those old plasma stored samples to predict cancer risk in the next generation. We're going for a Grand Challenges Cancer Grant as a part of a big team this fall. So that's kind of my new also interest.
Yeah. So in your lab, you also focus on other things like MECP2, as we mentioned, and the related disease Rett syndrome, and also other genetic neurodevelopmental disorders that involve epigenetics, like the Prader-Willi syndrome. But in the interest of time, I just want to focus on maybe your more recent work on this. With regards to Prader-Willi syndrome, you looked at a non-coding RNA and how this affects the DiUrin rhythm. I just had to look up what the DiUrin rhythm is. How does, non-coding RNAs are like all over the place, and it's really hard to really dissect or pinpoint like what it really does. And so what did you found about the connection of this non-coding RNA?
Yeah, this is probably way more complex to cover the time we have, because I've been working on this locus, over many decades. But Yeah, the Prader-Willi Angelman locus, this imprinted region, is kind of like a neuronal operon where all the parts are kind of regulating each other. But the non-coding RNAs in the Prader-Willi locus they're these, the minimal deletion region that causes Prader-Willi syndrome is these SNO RNAs, small nucleolar RNAs, but they are processed from within a larger non-coding RNA that's called the host gene. And we've seen previously by RNA fish that this host gene actually stays at the site of transcription, and it's like a cloud. It looks like the exist cloud on the inactive X chromosome, and it kind of acts.
But that is
similarly, although it seems to be more activating than repressing. So,
but the locus is not in the nucleolus then.
Well, the SNO RNAs go to the nucleolus. They're processed out and they go to the nucleolus, but the host gene stays at the site of transcription. And you get this chromatin decondensation that we've seen way back too. Anyhow, we've been working on this locus forever, but then more recently, as we started to get in the genomics and seeing the genes that were different, we saw that there were a lot of circadian genes that came up, as well as metabolism, like mTOR pathway. So it got me interested in metabolism, and it's really more the diurnal. So circadian genes are really, it's genetic, right? So if you put any organism in the dark, the circadian clock continues to cycle. But as you know, when you travel, you get jet lag. And so you can re-entrain your circadian clock based on the environment. So really circadian entrainment is the gene environment interaction. And so we'd seen this previously, that really doing RNA-seq at different times of the day in our Prader-Willi model, we got very different results. So that it was mostly like we saw the dysregulation of genes mostly in the middle of their sleep and in the light period. And then when we compared both the transcriptome and the methylome, one of the top terms we saw was circadian entrainment. So that's why it prompted us to do the current experiment, which is still ongoing, where we're actually entraining the mice to, instead of 12 hours of light and dark, we're going to 11. And then studying the effect of both the deletion and the overexpression of the SNORD116 locus. And that's really interesting. We're getting, it's a lot of data. And then we're correlating this back to the phenotypic data, which is like metabolism and as well as running wheel data. So that's still ongoing. But I'm really interested. The Prader-Willi Angelman locus evolved within mammals. It's not there in marsupials, and it's not there in the tenrec, which is supposedly the oldest placental mammal that's the most ancient that's been sequenced. So it is fascinating that this may be part of sort of a mammalian, mammals finding their niche in terms of sleep and metabolism, and they all kind of have. So that it's a fascinating locus for a number of reasons.
So what kind of effect does this cycling of this non-coding RNA have on the disease? So is there a type of intervention that you could do based on this?
Yeah, it's a good question. It is possible there could be, there's this field of chronotherapy where, either, adjusting people to the right time of day. Kids with Prader-Willi do have sleep problems. They fall asleep during the day a lot, like during school, but then at night they wake up more frequently. So yeah, it could be, maybe matching them or even just letting them go by their own clock. They might just follow their own time, and if school schedules could be adjusted or something for them. And in these cases, it could be, improve their at least health and well-being.
At least their mental well-being, if they're not pushed to something they don't like.
Yeah, and the problem is they're always hungry, so they're always seeking food. And again, that's another part of the whole metabolism issues that are going on with these kids.
And last but not least, you and your team profiled the gut microbiome and metabolome across disease progression in Rett syndrome, now switching over to MECP2. In the last couple of years, the evidence of the connection of the gut and the brain have stacked up. So there have been found multiple connections between the two areas. And this seems to go into the same direction. So what did you find here about the connection?
Yeah, and I've gotten convinced too, I guess, seeing the whole field progress. And it was interesting because usually you think about that as being environmental, but there are definitely genetic causes as well too to changes in the gut microbiome. And we've known for a while that, although you think of Rett syndrome as a disease of the brain, that many of the other body systems are affected. The girls have, range between constipation and diarrhea. There are a lot of GI problems. They have breathing abnormalities. They have all kinds of other, systems affecting the body. So we were interested, and what we have is I'd say 90% of the papers in the field of Rett syndrome are studying the knockout male mouse model, but it it affects girls in humans. It's an X-linked dominant disorder, and it's one of the few X-linked dominant disorders out there, right? There's probably a handful that you can count. And that for a good reason, because usually if you have a mutation on the one X chromosome, the other X chromosome is compensating for it. But in Rett syndrome, it's not. But again, they don't develop symptoms that you can't tell at birth if a child has Rett syndrome. They start to develop changes in their milestones around 6 to 18 months of age, and then it goes through a period of regression steps. In the mice, we don't necessarily see regression per se, but we do see in the female mice, we see a nice progression of symptoms, and it's both metabolic as well as motor, the primary phenotypes we see. So we wanted to kind of look at different time points to see the progression and how the females were different from the males. And the striking thing is that from the microbiome and the metabolome, we saw very different progression in the males versus the females. And it wasn't just that the females had, only one. It wasn't just the dosage of the mutation. And there was a lot of dynamics going on in the microbiome. And the metabolome, we saw butyrate came up before any of the symptoms. And that's usually one that's, thought to be a good guy. But I think in Rett syndrome, everything is a little bit odd. And when we looked at the lipids in the fecal pellets versus the brain, we saw some correlations there. So it's almost like they're not absorbing the lipids from the gut very well. And that could be a combination of the inflammation, the altered gut microbiome. So we are really now kind of interested in getting interested in lipids, dietary lipids, and how the gut absorbs the lipids in this model, because that could be a good avenue for therapy.
Because the neurons wouldn't develop well without the right lipids?
Right. So we really see deficiencies in lipids in the female brain that are correlated with what's going on in the gut.
So my last scientific question is you kind of touched upon this, but what are you working on right now? And let's say for the next 5 years, what are your goals that you are striving to?
Yeah, I think I touched on our NHIP project, which we're excited about is it neuroprotective and figuring out how it's regulated. So that's definitely one angle. I'm also, so we're getting really focusing on preventative types of care. So we've got a new center called the Perinatal Origins of Disparities, with the idea of using sort of perinatal life, again, epigenetic signatures from newborns to try to predict risk and prevent future problems, and also interventions during pregnancy to try. We're involved in collaborating with a social scientist now who's doing this mobile health intervention during pregnancy. And we're getting samples sort of before and after the intervention to look for epigenetic signatures. So it's that kind of thing. I think we're doing a lot more collaborations and getting more into the maybe more public health, precision public health and precision medicine so much. So,
I think for future grants, it's also important to have some kind of this in your science, right?
Yeah, I think I find it rewarding. I think I reached the point in my career where it's like I can either go where everybody else is going, which is, gene therapies for very rare diseases. But when you think about the societal impact of that, a lot of these families might not even be able to afford these therapies. Whereas if you could do something more towards, the more common disorders or preventative healthcare, I think that's kind of where I feel more rewarded.
So for the last 40 minutes, we have been on a journey through your scientific career. So did we miss anything important that you would like to add?
Yeah, I think we covered most everything. Yes, I think.
To finish off, could you maybe give us a brief summary of your most important finding, or that you would consider your most important finding?
Oh, the big one. Well, I think, I don't know if it's one finding, but I think it's, if I can kind of appeal to the young scientists out there, is kind of follow your nose in your research. I think that's what we've done. And I guess I'm just a constant learner, and we're always trying to fit out. Some people will tell you to focus completely on one molecule, one thing, and that is good advice. But I think if you do like to sort of follow your nose on research, don't be afraid to learn new things. Again, we've gotten into so many new fields. I didn't know much about the placenta at all until I, was introduced to it, and they were like, "Oh yeah, this actually could work for this." So be open to new ideas, to new methods, and to new collaborations. I think, and take them where go where they take you. And I think that's how we got to some of the discoveries that I'm proud of, like the discovery of NHIP and the discoveries in the Prader-Willi locus. We've kind of followed our nose on that one for over the years and continue to find it interesting.
So thank you, Janine, for your time and for being on the show.
Thank you.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.